Platelets, also called thrombocytes, are a component of blood whose function is to react to bleeding from blood vessel injury by clumping, thereby initiating a blood clot. Platelets have no cell nucleus. They are fragments of cytoplasm that are derived from the megakaryocytes of the bone marrow or lung, which then enter the circulation. Circulating and activated platelets are biconvex discoid 2 to 3 micrometers in greatest diameter. 6. Activated platelets have cell membrane projections covering their surface. Platelets are found only in mammals, whereas in other vertebrates, thrombocytes circulate as intact mononuclear cells. The ligands, denoted by letter L, signal for platelets to migrate towards the wound. As more platelets gather around the opening, they produce more ligands to amplify the response. The platelets congregate around the wound in order to create a cap to stop blood flow out of the tissue. On a stained blood smear, platelets appear as dark purple spots, about 20% the diameter of red blood cells. The smear is used to examine platelets for size, shape, qualitative number, and clumping. A healthy adult typically has 10 to 20 times more red blood cells than platelets. One major function of platelets is to contribute to hemostasis, the process of stopping bleeding at the site of interrupted endothelium. They gather at the site and, unless the interruption is physically too large, they plug the hole. First, platelets attach to substances outside the interrupted endothelium, adhesion. Second, they change shape, turn on receptors and secrete chemical messengers, activation. Third, they connect to each other through receptor bridges, aggregation. Formation of this platelet plug is associated with activation of the coagulation cascade, with resultant fibrin deposition and linking. These processes may overlap. The spectrum is from a predominantly platelet plug, or white clot to a predominantly fibrin, or red clot or the more typical mixture. Some would add the subsequent retraction and platelet inhibition as fourth and fifth steps to the completion of the process, and still others would add a sixth step, wound repair. Platelets also participate in both innate and adaptive intravascular immune responses. The platelet cell membrane has receptors for collagen. Following the rupture of the blood vessel wall, the platelets are exposed and they adhere to the collagen in the surrounding connective tissue. Low platelet concentration is called thrombocytopenia, and is due to either decreased production or increased destruction. Elevated platelet concentration is called thrombocytosis, and is either congenital, reactive, or due to unregulated production, one of the myeloproliferative neoplasms or certain other myeloid neoplasms. A disorder of platelet function is called a thrombocytopathy or a platelet function disorder. Normal platelets can respond to an abnormality on the vessel wall rather than to hemorrhage, resulting in inappropriate platelet adhesion/activation and thrombosis, the formation of a clot within an intact vessel. This type of thrombosis arises by mechanisms different from those of a normal clot, namely, extending the fibrin of venous thrombosis, extending an unstable or ruptured arterial plaque, causing arterial thrombosis, and microcirculatory thrombosis. An arterial thrombus may partially obstruct blood flow, causing downstream ischemia, or may completely obstruct it, causing downstream tissue death.